biology, cells and structures. And prior knowledge is what you should have learned from your Key Stage 3. That's the years in 7, 8 and 9. And what we should know is, what we should recall is that all living organisms are made of cells, basic building blocks of cells. Recall and identify some structures that are common to animal and plant cells. That means things inside the cells that are found in both animal and plant. Finally, recall that most cells are so small that they can only be seen through a microscope. So what you need to be able to do by the end of this, building on what you've learned in Key Stage 3, is to identify differences between eukaryotes and prokaryotes with examples. Name and describe the function of structures that are unique to animal, plant and bacterial cells. And finally, you should be able to describe the advantages and disadvantages of two different types of microscopy. Most exam boards talk about two different types. You should be able to say why they're useful and why they're not so useful. Okay, two types of cells. There are eukaryotic cells and there are prokaryotic cells. On the left, there are diagrams of eukaryotic cells, and on the right of the screen, there are prokaryotic cells. Eukaryotic cells are any type of cell that contains chromosomes stored in a nucleus that's surrounded by a membrane. The key idea at GCSE here is a eukaryotic cell has a nucleus. Prokaryotic cells are any type of single cell or single-celled organism that contains no defined nucleus or even defined chromosomes. The genetic material is just floating free inside the cytoplasm. So as you can see from the diagram there, the genetic material is the dashed circle that's kind of folded in on itself. That's not contained inside anything, it's just floating free in the liquid cytoplasm part of the cell. Animal cells. Animal cells are eukaryotic. They have chromosomes contained in a nucleus, and they have other subcellular structures. Animal cells also contain the following structures: cell membranes, cytoplasm, ribosomes, and mitochondria. Now we'll get into the function of these later, but you should know some of the functions of these, the jobs that they do, just from your studies and from what you've been taught in year 7 and 8. Plant cells, they're like animal cells again. They're eukaryotic. They have chromosomes contained in a nucleus. That's the key thing here, the commonality between both of them. But they also have other subcellular structures. Plant cells contain everything that's found in an animal cell, so everything that we listed earlier. That means that they contain a nucleus, they contain mitochondria, they contain cell membrane, and they contain cytoplasm. But they also contain some structures that are not found in animal cells that are useful for plant functions, such as photosynthesis, that animal cells don't have to undergo. Chloroplasts, a large storage vacuole, and a cell wall, which provides structure and support. So, animals versus plant cell then. We've mentioned these structures. We've got a diagram there on the left, and that's an animal cell. Diagram on the right, that's a plant cell. But what are the organelles and what are common to both? The cell membrane, it's the surface around the outside of the animal cell that controls what goes in and out of the cell. It's actually slightly different in a plant cell, in that that membrane is the inner layer around the inside of the plant cell. It's really important when labeling that you make sure that your arrowhead points to that inner layer and doesn't just stop at the outside of the cell, because that is a different layer altogether. A nucleus, this structure here and here, and that's where all the genetic information for the cell is stored. Ribosomes, as represented by the yellow dots here and here, these are important as protein synthesis. They are the site of protein synthesis. Mitochondria, the yellow structures there and there, these are the cell's powerhouses. There's where aerobic respiration occurs, and they're what release energy in the cell from the reaction between oxygen and glucose. Cytoplasm, that's the jelly-like liquid that fills the cell, and it's where all the cell's reactions and nutrients are stored. Then there's organelles that are only found in plant cells. Again, these are because plants do certain functions that animal cells don't need to do. Cell wall, that surrounds the plant cell and it keeps the structure rigid, preventing the cells from collapsing on themselves when there's lots of them together. This is in replacement of a skeleton, which animals have, so they don't need a cell wall because their support is provided by the skeletal structure. A large storage vacuole, this space here in the middle, that contains cell sap and is a store of nutrients, and it also provides a bit of turgidity to hold the cell structure in place. Finally, chloroplasts, these green discs here that contain a pigment called chlorophyll. Chlorophyll takes sun energy from the sunlight, and it uses it to drive the reactions that provide energy from sunlight. In other words, it drives photosynthesis. Bacterial cells, they're not eukaryotic, they're prokaryotic. They have no nucleus. Eukaryotic cells tend to be multicellular. There are lots of them in the organism. Bacterial cells, prokaryotic cells, tend to be unicellular. They have no nucleus, but they are single-celled organisms. Because of this, they're very, very small, too small to see with the naked eye. And in most cases, they're very difficult to view with a standard light microscope, like the one that you may have used yourself in the lab before now. To make them better to see, we can increase the magnification of the light microscope by using better and better lenses. Or we can use stains, which stain the structures and make them more visible. But even with a really good light microscope setup and a good stain, we can only really see the surface details of bacterial cells because of how small they are. Bacterial cells also contain the following structures that are not generally found in a plant or an animal cell. It's what makes them unique. Chromosomal DNA. Normally we find chromosomes in eukaryotic plant and animal cells, but in bacterial cells, the chromosomal DNA is just found floating, kind of looped on itself in the cytoplasm. Plasmids. Plasmids are circular pieces of extra chromosomal DNA. That means it's not necessary for the organism. You don't always have it. But that plasmid can contain genes that can confer an extra advantage on it. So for example, if that particular bacteria is resistant to an antibiotic, it might be that that resistance gene is in the plasmid and that it's acquired that from somewhere. A slime coat. Some bacterial cells have slime coats around the front of the cell wall, and the reason for this is, one, to make it easier for movement, but two, also provides protection against defense mechanisms that the bacteria might encounter. And flagellum. This is a long whip-like structure, which is a bit like a tail, if I'm honest, and it's used to propel the microorganism, the bacterial cell, through whatever medium it's in its environment. So it's basically like used for swimming. So this is our bacterial cell. You can see that they've got ribosomes, a bit like plant and animal cells. The ribosomes in bacterial cells, though, are actually smaller than eukaryotic ribosomes, but they do the same job. They are involved in protein synthesis. We also have a cell wall. Again, it's on the outside. The inner lining, where the grey meets the blue, would be the cell membrane, as evidenced here. Again, you have to be really careful if you're asked to label a diagram of this to get those arrows in the correct position. Because if you mix it up, or if you're out and it looks like your arrow for your cell membrane is on the cell wall, or your arrow for the cell wall is on the cell membrane, you can lose easy marks. Chromosomal DNA, this dashed line here, it's looping in on itself, which is the DNA found floating freely just inside the bacterial's cytoplasm. Plasmid DNA, these circular pieces of DNA that contain extra genes, should I say, not chromosomes, that can confer advantages on the bacteria. The flagellum, the long whip-like structure that it uses to move through its environment. Cytoplasm, again, just like eukaryotic cells, it's a nutrient-rich medium, jelly-like medium, provides support for the cell to keep it turgid, but also contains dissolved substances. It's where most of the chemical reactions of the cell occur. So that's the differences between eukaryotes and prokaryotes with examples. And also now we're going into name and describe the function of structures unique to animal, plant, and bacterial cells. So what do the structures actually do? I've touched on them, but in the interests of knowing that this is being recorded for you to view for future, I figured that the best thing to do with this is to have this written down in case you don't have time to make notes, so don't panic too much. You can see this in future when you watch back. Cytoplasm. It's a jelly-like structure containing dissolved nutrients and salts, and structures called organelles. It's where most of the chemical reactions for any animal, plant, and even bacterial cell occur. The nucleus. Only present in eukaryotic cells, like animal and plants, not present in bacterial. They contain genetic material, including DNA, chromosomes, genes, that kind of thing. The cell membrane. This controls the movement of substances in and out of the cell by being permeable, should I say, to some substances and not others. That means it'll let some things through, but it's selective. It won't let everything through. It's a bit like a gatekeeper or a doorman on a door. Mitochondria. Organelles where respiration occurs and where most energy is released in respiration. This is where the glucose and oxygen is combined to release energy, and carbon dioxide and water is generated as a waste product. Ribosomes. These are tiny structures in plants and animal cells where protein synthesis occurs. This is taking amino acids and combining them to make long-chain amino acids, which then are used to make proteins. Then there's the structures that we mentioned that are only found in plants. They're unique to plant cells. Chloroplast. It's an organelle that contains a green pigment called chlorophyll, which is needed to absorb light energy to drive photosynthesis. It's the majority reason why leaves on plants are green. It's because they contain lots and lots of chloroplasts with lots of chlorophyll, especially near the surface of the plant leaf. If you take the chlorophyll out of a plant leaf, or if you deprive plants of certain nutrients that it needs to produce the chlorophyll, the colour of the plant's leaves will change from green, and you've seen this probably yourself in the wintertime, from green to red to orange to brown. And when that happens, the plant can't produce energy from the sunlight using photosynthesis. Cell wall. Plant and bacterial cell walls provide structure and protection. The plant walls are made from a material called cellulose. Large permanent vacuole. This is filled with cell sap to help keep the cell swollen or turgid. And animal cells can contain small temporary vacuoles, but with plant cells, it's a large and permanently present vacuole. So what functions do we have for the structures that are unique to bacterial cells? We mentioned some before. Chromosomal DNA. The DNA of a bacterial cell is found loose in the cytoplasm. It's not contained in a nucleus, and so because of that, we call it chromosomal DNA, not a chromosome. Plasmid DNA. Small closed circles of DNA that can give rise to variation, such as an antibiotic resistance in bacteria. Plasmids can move from one bacterium to another. We say that you can infect a bacteria with a plasmid, but the chromosomal DNA can't do that. Plasmids seem to be able to move, chromosomal DNA stays put. Flagella, which is the plural for flagellum. Bacteria have more than one flagella, singular is flagellum. These can move in a whip-like fashion or even rotate a bit like someone spinning a chain in the hand to help propel the bacterial cell forward through whatever medium it's in. And then finally, the cell wall. This helps provide structure and protection, but they're not made of cellulose like a plant cell wall. In bacteria, they're made of different materials. So that helps name and describe the function of structures unique to animal, plant, and bacterial cells. And that's something that at GCSE, if you're asked, it is pretty much just a case of learn it, because if you can recall that and put it down, you know you're on a winner. You've got some easy marks there. Final point, describe the advantages and disadvantages to two different types of microscopy when viewing cells. So what we've got here are two different pictures. We've got a light micrograph, which is a picture taken under a microscope, and we've got an electron micrograph. The light micrograph is on the left, the electron micrograph is on the right. The difference in the detail of the two images, both are from microscopes. The light micrograph on the left shows some of the structures that we've encountered in this lesson. You can see that it's got labels for chloroplasts, the vacuole, the cytoplasm, the nucleus, and the cell wall. But there's very little detail to those structures, and the images are very, very small and very fuzzy. The electron micrograph on the right shows a lot more detail. We can make out individual features of these structures. So if you look, you can see that the chloroplasts have that kind of banding pattern on there. You can see the nucleus, and you can see the nuclear pores that are present on the surface of it. You can see the cell wall, and you can actually see the thickness of the cell wall. So the electron micrograph allows us to see a lot more smaller organelles in a lot more detail, they magnify. Why bother using a light microscope then? Well, light microscopes, we're right, we do have limitations compared to an electron microscope. The best magnification of a light microscope, roughly, is around 1500 times, meaning we can make an object that's 1 centimetre appear 1500 times bigger, 1500 centimetres, with a resolution of about 0.0001 millimetre. Resolution is the smallest distance between two dots on a screen before it starts to look like you've just got one thing in front of you. It's the separation between the two. The better, or the smaller the number for the resolution, the smaller the gap you can see before it blends into one. This is including the use of optimal light sources and stains, which we mentioned earlier, to get the best image out of it. Compare that to an electron microscope. They have a best magnification of around 2 million times, with a resolution of about 0.000002 millimeters. They give much greater detail and images of greater sharpness or clarity. Those are keywords to use when you're using them in the exam. Sharpness means the actual image is not blurry. You can see details with greater resolution. And clarity, it means the same thing, but the resolution's key as well. Problem is, light microscopes have one advantage over an electron microscope. Light microscopes can be used to view a living cell. For an electron microscope to be used, we have to prepare whatever we're viewing, the sample, with a gold coating. If that's a living organism, then coating it with gold in order to see it under an electron microscope will kill it. So we can't use it to see a living organism or to see living processes occurring in real time, where we can with light microscopy. So a quick summary of these things. A light microscope, its main advantage can be used to view live cells or organisms, but its disadvantages are its max magnification and resolution is not high enough to view the smaller organelles with much detail or clarity. The electron microscope has the advantage of having everything right with it that's wrong with the light microscope. Its maximum magnification is much higher, and its resolution is so much better that it allows for images of very high clarity that show the smallest organelles in detail. Its major disadvantage is that we can't use it to view live cells. All the samples are dead because of the way they're prepared, so we can't use it to image living cells to see how they function. Incidentally, you get a 6-mark question on this or a 4-mark question on this, you can answer in this fashion with a table like this to help you structure your answer. It's a valid way of presenting answers that will be marked by the examiner. That's the final outcome. Describe the advantages and disadvantages to 2 different types of microscopy and viewing cells. That's the end of today's lesson. I hope you've enjoyed it. If you have any more questions, feel free to contact using the details at the end. Thank you very much. Have a nice evening.